Hi everyone, we're going to be taking a look at a lab that has to do with the breakdown of hydrogen peroxide, which is a toxin that your body makes, into water and oxygen, which are harmless. It requires an enzyme to do this. The enzyme's name is catalase. So what I have here is some yeast that I've put in water, and so it's kind of a mixture. And in here I have hydrogen peroxide, and it's hydrogen peroxide that you could get at the store, right? Same stuff. And we're just going to look and see what the reaction looks like. So this right here has the catalase in it, which is our enzyme. And when I pour it in, and you watch, you can see what happens. And as you get closer, you can actually see all the bubbles, right? You can see all the bubbles. That's the oxygen coming off, and that's the oxygen that would be going into the balloon. And once that oxygen is in the balloon, then we would measure how much is produced. And you can see that the longer it sits there, the higher the bubbles go. And we could have actually measured the height of the bubbles after a certain amount of time. That also would have helped us. There are all kinds of different ways of measuring this reaction, but we're going to be using the lab that uses the balloons. In this particular lab, the scientist has put hydrogen peroxide into a test tube and put the test tube in a beaker of water of a particular temperature, which is why we have the thermometer there. Into the test tube, he just placed catalase enzyme and then put a balloon over the top of the test tube that would capture the oxygen. In this case, we will not be measuring the product water. We're going to be measuring the oxygen bubbles. As the reaction is taking place, oxygen is going to come off of the reaction because it's the product, and it is going to fill the space in the balloon. After 3 minutes, the scientist will remove the balloon from the test tube and measure how much oxygen is in there. The scientists set up 5 test tubes at 5 different temperatures, and you can see all of the constants for each setup. We can't actually do this experiment, but we have been provided with the data, which you are going to need to average because the scientists did this experiment 3 times. So for instance, for 10 centimeters, you're going to find the average. To find the average, first you're going to add together the 3 trial measurements. So that's going to be 6 plus 9 plus 7, which happens to be 22. And we're going to average it by dividing by the number of units that we actually added together, which is 1, 2, 3. So we're going to take 22, we're going to divide it by 3, and that means our average is going to be 7.3. Now you're going to do the same thing for 20 centimeters, 30 centimeters, and 40 centimeters. On your lab, your 50 centimeter has been done for you because all of those measurements are the same. The average is going to be the same. Once your averaging is done, you are then going to plot your points on your graph. We're going to be making a line graph here. And you're going to have 1, 2, 3, 4, 5 points, 1 point for each of our temperatures. So you will be graphing 10 centimeters with whatever your average is, 20 centimeters with whatever that average is, and so on. Remember that your IV goes on the x-axis and your DV goes on the y-axis. Make sure that you have completed the scales on both axes and you have labeled them, and that you have also done your title. When you're done plotting your points, you are going to need to connect the dots so that you can see the pattern of enzyme action. Once your graph is done, complete the analysis questions, and then your completed document should be submitted to the assignment on Schoology.